Welcome to The Edge, a program where we spotlight Purdue University faculty, scholars, and innovators, highlighting their careers, scientific achievements, and their vision for the future of their scientific field. My name is Tommy Source. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology and Infectious Disease. And we continue our focus on microbiome sciences today. I have the pleasure of welcoming Dr. Timothy Johnson, Assistant Professor in the Department of Animal Sciences at Purdue University. Welcome to the program, Tim. Thank you so much for joining us.
All right. Thanks for inviting me. This is going to be fun.
This is going to be great. You know, I've been wanting to talk to you because I think your work is so relevant for so much going on in the world of health today, both animal health, human health, and everything that relates to antimicrobial resistance and all the work that you do in that capacity. I wonder if you could take us back a little bit. Tell us about your background, what was it that led you down this path? And maybe we can start there and go from there.
Well, how far do you want to go back?
As far as you want to take us. I mean, I've seen some of your earlier publications that you've posted on your profile. And I don't know if you want to take us back to some of that work where you started to get into, microbial ecology and, intestinal microbiome and all the work that you started to do, at least the first few instances, if you can tell us the inspiration there.
So I'll take you a little bit farther back, I think. Maybe for those that are listening, sometimes our path is a little bit not linear. So I'll just briefly, my undergrad, I changed majors every year. First, I was a math major, math education, because I like math.
Yeah.
And then I got frustrated in calculus 3, and I don't know. Then I wanted something more, real world. So I completely changed to landscape management and just construction, which was completely different, right? But that I enjoy nature and outside, and I am interested in landscaping and that kind of thing for fun. But that wasn't really what I thought was a good career choice. But in the course of that, I took a soil science class, and I was blown away by the importance and impact of soil microbes and soil processes and cycling carbon. Global carbon and nitrogen cycles are controlled largely by soil microbes, which I hadn't ever considered in my whole life. So that started me on a track in soil microbiology. And then I had the privilege to go to Michigan State to do my PhD under Jim Teege, who's kind of a founding leader in the field of microbial ecology. He founded the Center for Microbial Ecology at Michigan State. And so wonderful, wonderful experience to rub shoulders with all the students that went through his lab and with him. So there, I guess I got my first starts. I was studying antimicrobial resistance in animal agriculture systems. Interestingly, as a side project, that was my first kind of just test project to see how I would work. And he was testing me to see what I was willing to do. So
like all good professors do.
So that was really interesting. We quantified antibiotic resistance genes in animal manure, and also as manures are applied to soils, if the abundance of those genes increase or decrease, or how that works in the actual real world setting.
Yeah.
And we found that, in animal agriculture, due to the use of antibiotics, there is a selection for antibiotic resistant genes, antibiotic resistant bacteria, which is really important, because the animal industry is large and their impact is also large. And so we need to be aware and trying our best to make the best choices possible. So that was my start, and I think was a wonderful PhD. And then I went on to Canada. I know you're a Canadian by birth. So I went to Ontario, to McMaster University,
Yeah.
to then try something completely different, which was I joined the lab of Jerry Wright. He's a biochemist and studying in a similar vein to what I had been doing, but different was he's focused more on the synthesis and production of antibiotics and discovery of novel antibiotics that could be new therapeutic drugs, especially focusing on how to combat antibiotic resistant bacteria. So anything from identifying inhibitors of antibiotic resistance genes so that more traditional antibiotics can kill them by blocking their resistant mechanisms, or discovering completely novel antibiotics, or repurposing previous antibiotics. So that lab was wonderful because it was very mechanism based, trying to discover exactly how things work, which in the ecology field, sometimes we don't worry so much about exactly how things work, but trying to observe how things happen in natural settings. So I think that was a great training for me. And then let's see, and then I went to the National Animal Disease Center in Ames, Iowa for a second postdoc. And there I got a lot of animal training, of running actual. Up to that point, I hadn't done an animal experiment, but we actually raised the animals, sampled, took intestinal samples, and also then looking at the microbiome host interaction.
Okay.
So
Interesting that yeah.
those three things kind of I think formed a good background for me, both having a strong ecology background for those principles, and then focusing on mechanisms, and then also how to actually perform experiments in animals. So those three together, I think, were very helpful for me.
Absolutely. I think that convergence of those three really big training branches, right? They're really separate modalities that you've combined into your lab and into your program. Now, it's so amazing to see that trajectory. I was born in Venezuela, but I did move to Canada at an early age. But just to be clear, I didn't want anybody to be misled. But definitely, what a journey through these different labs to learn these different techniques. There's so much that I'd like to unpack there if we can. Your perspective on your first inspiration in looking at the soil microbes and having such a huge impact on cycles that occur on the planet, and then having that capacity to then go in and start to understand how these soils get I guess changed, so that they become changed by the animals that we grow on them, the plants that we grow on them. And again, like, tell us a little bit more about this connection between the animals and the soil microbiology, if you can.
Okay. The soil microbiology is just well, Dr. TG likes to compare the microbial ecology to astronomy, or space. I don't know what the correct term is, space science, which is kind of a very different thing to compare soil microbes to, but with the idea that there's so much unknown in the universe. We can discover new planets, new solar system, new I'm extending the edge of my vocabulary. But it is so expansive and
Yes.
largely unknown.
Yeah.
And the similar things can be said in especially in soil microbiology. The diversity of bacteria in soil is so vast.
Mhm.
Probably one of the most diverse environments on Earth. And there's a lot of different processes going on, a lot of different competitions. Mostly soil is nutrient poor, and bacteria are just struggling to survive, or oftentimes going dormant and just hanging out, waiting for, that next rainfall event, that next dead something to give them nutrients. But really categorized by huge diversity, huge potentials, and waiting around. So when we mix animal waste or it's mostly animal waste getting mixed in with soil.
Sure.
That's one of those events where things start to happen, providing nutrients, providing moisture, and then this immense diversity has the ability to start performing functions that maybe they've been doing very, very slowly up to that point. And so for me, it's one of those gradient type or change events where interesting things can happen. So for me in particular, when we're introducing, so it's something that we're trying to capture a little bit more right now in my lab in a small way, is so the animal waste came from a largely anaerobic environment where oxygen is not present and the anaerobes are happily doing their functions. And then that waste is excreted and joins an aerobic environment. And then there's a huge amount of competition and change and turnover, and bacteria's innate desire to survive. And then you mix it with this other very diverse community in soil. So there's like a lot of things happening all at once. And you add into that also that there may be antibiotic resistant bacteria in there with resistance genes on plasmids or resistance genes that could help other bacteria survive. And so what happens at that interface too?
Yeah.
Can those plasmids can be shared to other bacteria through conjugation? Yeah. And so where are those plasmids going is a huge question.
Yes.
Which bacteria survive and which bacteria die?
Yeah.
And so it's at that, like, microbiology is very survival focused, right? And so they will do anything to survive. And so it's in those times where big events can happen. And for me, it's really important to understand if under these survival conditions, if antibiotic resistance is spread more broadly throughout a microbial community, or if those bacteria just die peacefully and we never hear from them again, which would be ideal, right? But
Right.
we want to be sure we know what's going on in that really intense dynamic that's going on.
Absolutely. So, I think if we can go back just a second so that people really understand what we're talking about here. So what you're saying is that bacteria can harbor antibiotic resistance genes, or genes that allow them to survive under antibiotic pressure, if you would like, and that these genes are not encoded in their regular genomic DNA, but the genes are on these extra genomic or extra other types of DNA, circular pieces of DNA that can be passed on from one bacteria to another. Is this how you're this junction of or this ability that bacteria have to transfer their resistance to their neighbor bacteria?
Yeah. I'm tempted to go maybe farther back than maybe I should, but I'm gonna do that anyway. I'm gonna go a little bit farther. So I'm gonna pull together a little bit. So in my time at McMaster University searching for new antibiotics,
Yes.
the vast majority, and this kind of brings a lot of things together maybe, and hopefully doesn't confuse too much, but the vast majority of our antibiotics, like if you ever heard of an antibiotic called streptomycin, or any antibiotic that ends in mycin,
Yes.
those all came from soil bacteria. They're produced by they're produced by bacteria that were isolated from soil because they had this property that they could kill their neighbors, their neighbor bacteria. So that's an incredible, powerful function that a bacterium can have to kill its neighbors, to be able to get those resources that it needs to survive in soil. So those same bacteria that produce antibiotics don't want to be killed by themselves. They need to survive because they're producing a toxic molecule. So they, by necessity, have to encode a resistance gene for themselves to not kill themselves. And so those genes evolved naturally, and have been around for millions and millions of years. But the interesting thing is, sometimes, like you were saying, then those genes can become shared. I can't share my genes with you, and under no circumstances, unless it's some kind of genetic manipulation science project.
Right.
But under natural conditions, we cannot share our genes with any other person except our children.
Yes.
But bacteria, they are not like us. They have their main chromosome, which they pass on to their progeny, but they also have accessory DNA, which can be shared, usually with closely related relatives, through this process called conjugation. So there are these little pieces of usually circular DNA called a plasmid. But there are many ways that bacteria can share their DNA. We won't talk about all of them, but bacteria can definitely share their DNA. Yeah. And when something toxic to them is present in the environment, say an antibiotic, the pressure or the occurrence of sharing this DNA increases dramatically, because it's about survival and they want their species to survive. And so all those survival mechanisms, it turns on this huge cascade in the bacterial cell, and they'll do anything to survive, including sharing DNA. So that's how a lot of these resistance genes can become more broadly disseminated between bacteria. So then the problem comes if those genes get shared to bacteria that could make us in in our selfish motivations, we don't want bacteria that make us sick to get these resistance genes. You know, if it's just soil bacteria, actinomyces or whatever that get these resistance genes, they're not really going to impact us in much of any way. But the thing that is really concerning is if these genes can be shared or acquired by animal pathogens or human pathogens, which then could create a huge problem for human health. So hopefully that's an interesting way to explain this. And
so interesting. You have no idea. You have no idea how interesting this is. First of all, I hope people out there are listening and hearing with broad ears that, you know, this the study of antibiotics and discovery of antibiotics takes us back to Ian Fleming and at discovering penicillin in a dish, just like how you said, with fungus growing around bacteria and the bacteria dying around the fungus. And like being able to visualize that and being able to take that in the 1940s when he got the Nobel Prize. It hasn't been that long, and it's such a young field where people like you are starting to really uncover this. You said something that blew my mind too, is the bacteria are fighting this war, this battle to kill off whoever might compete against them. But in doing so, they need to shield themselves from their own weapons that they're using to kill the other bacteria. And I think that's a huge eye-opener for people. The other huge eye-opener is that as we apply more antibiotics and we continue to pressure the bacteria and try to kill off the ones that are not antibiotic resistant, you said there's a higher propensity to transfer genes from the plasmids to create a larger pool, or to, you know, help the numbers continue to survive here. So can they transfer this to non-similar bacteria? So can this transfer happen from one bacteria type to another? And is this happening in our guts too, and in animals' guts as well?
So it's difficult to observe these kind of events. But so I can't exactly give you like the frequency of these things, but you asked a couple of questions. So how do the genes transfer? So there are I don't know what I can compare it to, but there are what are called narrow spectrum plasmids and broad spectrum plasmids. And so some plasmids are really very specific to probably one species, and they're shared only with their close relatives. But then there are broad spectrum plasmids that are more broadly disseminated. They're still probably not going to be shared, you know, there's still a range, you know. I don't think there's any plasmid that just can be shared with whoever they want. Right. There's large divisions within bacteria, but there's gram-positive bacteria, which are one major class, and then gram-negative bacteria, which includes E. coli, which are another big class. And so being able to transfer plasmids from gram-negatives to gram-positives, I think, is one of the least frequent things that happens. And we tend to treat these two groups of bacteria differently. So we do know that these kinds of types of plasmids exist, narrow and broad spectrum. But then I said it's difficult to quantify the frequency at which these events happen, because then if it just, even in my opinion, even small events could then lead to big consequences for a long time to come, right? It's if you transfer a new gene into a new species, it's been observed that usually we talk about selection pressures and the cost of carrying new genetic material. It's a general principle that bacteria will only carry the genes essential for its survival. And then if there are genes not required, that's going to be a cost on its lifestyle, on how much energy it needs to expend. And those that carry all this extra material, they'll be outcompeted by the more lean, more quickly reproducing bacteria. So these genes could be seen as a burden, right? Because they're carrying extra genetic material. But what's been observed is that it's actually not that big of a burden to bacteria to carry extra antibiotic resistance genes, because most of the genes are pretty short, like 1,000 base pairs, which is only a tiny fraction of their total genome. So what I'm trying to get around to saying is that the transfer of maybe a few transfer events could then lead, bacteria reproduce very quickly. So one transfer event could lead to one resistant bacteria, but in a few days, one bacteria could turn into millions of bacteria.
Yeah.
And so that's kind of sometimes what makes the observation of these events difficult, because, well, was there a transfer event, or did just some really rare members of this community that had these resistance genes just multiply? There's two pressures. There's a selection pressure to select for bacteria that are already resistant for them to grow and multiply. And then there's a pressure for this transfer event to transfer resistance genes. And it's difficult to disentangle which one is happening, or if maybe both are happening at the same time. So to get at it a little bit, we can quantify the genes that are responsible for transfer. We call them mobile genetic elements. So these are genes responsible for mobility of other DNA, like plasmids are mobile genetic elements. And so we can quantify how abundant mobile genetic elements are. And we see that under the use of antibiotics in animal agriculture, mobile genetic elements increase even more. From one of our studies, they increase more than the resistance genes themselves, about 10 times more.
Wow.
On average. So resistance, so that just is another sign that bacteria want to survive, and by sharing their genes is a key mechanism by which they can survive.
Yeah.
So you asked, does this happen in the gut? I think yes, definitely. There's a paper not published by us, but by a group from MIT, and they tried to look at all different environments and quantify genetic transfer events and like plasmid exchange. And they found that the gut, both animal and human gut environments, is where another term for this transfer is horizontal gene transfer from between living bacteria.
Yes.
So horizontal transfer is the most common in the intestinal tract.
Oh, interesting.
Which should kind of, it's I think why is because it's nutrient rich. There's a high concentration of bacteria. There's often selection pressures based on diet. And so when you get a bunch of bacteria together they're all crammed in this little tiny space. And they have to be able to touch to be able to transfer genetic material. And so that sets up a condition which is conducive for horizontal transfer of genes.
Right. And so what you were explaining before is that these mobility elements enhance the horizontal transfer. So have the studies been done where they knock them out and you reduce the horizontal transfer, or you have them or overexpress them and enhance the actual transfer?
Well, the genes are the ones that are responsible. They don't enhance. They're the genes responsible for the
Got it.
transfer.
I see.
So yeah, if you knock them out, then that type of transfer won't occur.
How interesting. How interesting. And Tim, tell me.
And I guess another layer of complexity. I love the topic. So
Do it. I love it too.
There's also transfer between bacteria, right? Largely through plasmids, but then there's also genetic rearrangement within one bacterium, within one cell. So bacteria can cut out a piece of their DNA and they can move it somewhere else in their chromosome, or recruit and put it onto a plasmid, or sometimes resistance genes there's actually a genetic element called an integron, which basically recruits largely, it could be any gene, but largely resistance genes, and it just lines them up into this series of resistance genes. Sometimes it can be 10, 20 resistance genes in a row. And then that whole piece could be cut out and put into a plasmid. And they've observed some of this. It's still difficult to observe these things happen, but it's been observed in some cases. So
there's genomic...
this kind of assembly of multiple resistance genes. And so these are the kinds of things that should terrify us. And I don't want to terrify anybody.
Yes.
Because, so I'll offer another perspective, is that actually a paper that came out from Jerry Wright, my postdoc advisor, I can't remember the title exactly. It's something like, the main idea is why are bacteria just not all completely antibiotic resistant by now? Because most of the times you take an antibiotic and it works. Most of the time.
Yes.
Antibiotic resistant pathogens are increasing. Sometimes you do hear horror stories when a person can't be cured by antibiotics because their infection is resistant to everything that they're giving the patient. But by and large, still, despite our last almost 100 years of antibiotic use now, antibiotics are still mostly effective. So I don't want to paint doom and gloom stories. Mostly awareness and trying to be careful, be judicious, and as wise as we can.
Yeah.
But there are really interesting potential for bacteria to become total. It's a new term, total antibiotic resistance, especially in tuberculosis. There's been this progression from drug resistant to multi-drug resistant to extended drug resistant to
superbug
to totally drug resistant.
Yeah.
So that's what we want to avoid.
Yeah, absolutely. And I thank you for taking us down that path. And I know for some it might be a little too molecular, but so interesting for me to hear about some of these, and fascinating to hear the transposable elements that can be transferred from genomic to plasmid DNA and all of that, and lining them up, resistance genes. I can just see like an operon lining up. But so Tim, let's move if we can now to the my gosh, the immense amount of work that you're doing in animal microbiome. You've been very active in swine microbiome, but I also see you now doing some respiratory, bovine respiratory virus as well work. And I see you now also looking at this connection with animal health and microbiome. Tell us a little bit, what's the world of swine feed and swine microbiome, swine health? Because I see that's been a big thrust of a lot of your work. I wonder if you can tell us a little bit about that.
Okay. So when I came to Purdue, I wanted to be, so I went through a bit of a transition. So it's useful, and I just talked about all the problems associated with antibiotic resistance. But so describing the problem of antibiotic resistance is important, and we need to know about it. But when I got here, I wanted to be, instead of telling everybody all the bad news, I wanted to try to be a part of a solution and find new methods, new management styles, new things, new feeding strategies that could be used to improve animal health without relying on antimicrobials. So traditionally in animal agriculture, antibiotics have been used to treat infectious disease, to prevent infections from circulating in a group of animals, or also to promote animal growth. So because of all those uses and the number of animals in the world, agriculture is one of the largest consumers of antibiotics in our society. And but they are, animal producers are just like all of the rest of us. They're doing the best they can with the resources that they have, and trying to raise healthy animals that are as happy and healthy as they can be. And so, but there have been recent changes to that situation. So there's been new legislation that disallows the use of antibiotics that are important in human medicine to be used as growth promoters for animals. And so that's taken some of their tools off the shelf that they can no longer utilize now. So there's a big focus now on what's the field of what's called antibiotic alternatives. So trying to get the same benefits that were provided by the use of antibiotics in other ways. Some of these are strictly dietary, trying to optimize their diets even further to optimize health. And others are trying to improve animal immunity and also gut function, and which is the microbiome. So where are we in? Actually, now I've forgotten your original question, but
about swine, and I guess, where are we with helping that industry? And I really, again, so informative, and I hope people kind of understand that there's such I mean, what would you say, 2/3 of the world's antibiotics are used in the animal industry to maintain those populations healthy, right? As interventions, as you said, potentially.
Those numbers are debated.
Yeah.
But
But it's a large amount.
There's a large amount.
There is a large amount. So, but what's the risk? Are we consuming the antibiotics when we consume the flesh of those animals, or is it mainly, as you said before, that the animals' microbiota that comes out in the waste is causing the problem further down the chain?
Okay, so yeah, maybe this is a little bit of myth busting
Good.
section.
I love it.
The use of antibiotics in the United States, I can't say for every country in the world, but definitely in the United States, the use of antibiotics is tightly regulated. Antibiotics have to be used as described. There are required withdrawal periods so that when antibiotics can be administered either in feed or through a shot into muscle or subcutaneous. So if antibiotics are administered in muscle, that antibiotic is then distributed throughout the body, and there's a required time which has been shown to eliminate the antibiotic residue from meat. And so there's there are those rules because we know that we don't want to have antibiotics in the food that we eat. So that would not be good. So I'm not too concerned about antibiotic residues in the food that we eat. I don't think that's a widespread problem in the United States. The animals themselves do not become antibiotic resistant. It's the bacteria in the animals that can become antibiotic resistant. Those genes are not transferred from bacteria to the animals. So don't have to worry about that. It's largely the bacteria in the intestinal tract that are where the antibiotic resistance problem comes in. So as long as you're cooking your food thoroughly, you're being sanitary, your risk is pretty low for direct consumption of antibiotic resistant bacteria in your diet. This has been quantified by others in our department. So basically you can think about it this way. What is your risk of food poisoning from eating meat? Food poisoning does occur, and usually that's because there's some kind of contamination from feces or intestinal contents that would contaminate meat during the slaughter process, and that can occur. Salmonella, E. coli, there are contamination events. So there's a certain, risk of that. I don't get food poisoning that often. So that's fairly rare. And then you have to think about, well, an antibiotic resistant food pathogen is even more rare, right? Because so basically you got this food illness risk and then a antibiotic resistant food pathogen, which is even smaller. So that route is, I think, fairly small risk. So that's not what keeps me worried. It's the more direct routes of what's happening to animal manures. Where is it going? I recently, so those animal manures could go into water, surface water, rivers, streams. It ends up in soils. It can end up in dust that gets blown from potentially over large distances. It could go into groundwater, these bacteria. And so those environmental paths may not come back to the human population very easily, but for me, that's where the vast majority of the resistant bacteria go. But then circling back and getting back to humans, at that point, there's a lot of dilution going on, and so the risks are also go down. So, but there could be, potential small events, which then could lead, like I said earlier, some small events could lead to bigger impacts. Probably something that really kicked off this whole field of studying resistance in animals was way back from the 1970s. There's a researcher from Tufts University, Stuart Levy. He's a medical doctor, and he was looking at infections among farm workers and their families at poultry farms, and if those infections were antibiotic resistant or not. And so from that work way back in the 70s, he found that farm workers and their families were more commonly colonized with antibiotic resistant bacteria than neighbors in their communities. So there could be potential, occupational risk in those kind of careers. But I think as that has become more aware, the modern farm is nothing like the farm of the 70s, and there are so many controls, and I think they're improving every year. So but it's something to be aware of and something to minimize, and definitely the animal industry is doing their utmost to try to be as responsible as possible.
Yeah, it's so interesting, this mention of transfer also of diseases from animals to humans, and how that impacts the industry. I know, for example, bird flu, avian flu has been going around, and that's also of a concern. Tell us a little bit about this respiratory, bovine respiratory disease, yeah, and a little bit of that work. I mean, it's so interesting, that collaboration. And Tim, if you can, in that answer, if you could tell us, who are you collaborating with, and what are the typical types of collaborators you have in your work?
This is a great project. So it's a large project. We have funding from USDA. So this project involves myself as a microbial ecologist. I'm focusing on, on the microbiology side, and then we have the lead is from ag and biological engineering. And then we have also a dairy specialist, beef specialist, extension. We have people in communications conducting surveys of producers and veterinarians. And so the whole goal of this project is that bovine respiratory disease is essentially just like us, upper, lower respiratory infection. We would call it the cold, bronchitis, pneumonia, runny nose. All of that falls under this collective term of bovine respiratory disease in cattle. And they have the similar symptoms. They're tired, they don't want to eat, they want to lay down, they get discharge from their nose, sometimes discharge from their eyes, and they're sick. And the farmers don't know why, right? Just like us. Is it a cold? Is it the flu? Is it bacterial? Is it viral? Is it pneumonia? Is it bronchitis? Like, where is it? And they're it's a huge struggle because they have many animals, and so you can't spend hours on each and every individual, and they have to make quick decisions, and there's a lot at stake. Those animals are large, expensive animals, and so as a result of this, um, the the current practice is they will use the best knowledge they have available, probably based on treatment successes in the past. Maybe they have somewhat of an idea of what might be circulating on their farm, and they will either choose to um, provide supportive care. Um, uh, Sometimes they'll provide anti-inflammatory drugs, or they'll put them in a sick pen and try to get them more water. They'll give veterinary exams to try to see what's happening. And then in the worst case scenario, they'll choose to administer antibiotics to try to clear this infection. But kind of what spurred this on is a lot of time, about 30% of these antibiotic treatments are not effective, either because the bacteria are resistant or maybe because it's a viral infection and not a bacterial infection. So our goal in this project is to provide rapid information that can be used by the farmers in the field. Currently, you might have to take a sample and send it to a veterinary diagnostic lab, which may be some distance away, and then wait for them to culture and to do this work. And that may take days or weeks, but they need to make a decision now, right? So that's where this collaboration comes in. Our partner in engineering, he's working on developing a device that could provide a rapid test, kind of similar like we've all been taking COVID tests. It's not exactly the same test, but the same principle. Take a swab of the animal's nose, put it in a test, and try to get some answers. We are working on trying to develop, this test is a little bit more complicated because we're not just looking for COVID-19. We're looking for a whole host of bacterial pathogens, which could be the cause. And basically going from how do we interpret the results on the test to what that actually means for the animal, because that's not exactly clear cut.
Yeah.
And so it's a great project. For me, it hits at trying to use antibiotics judiciously, hopefully with this kind of rapid information.
Yeah.
The best choices can be made right at the time of needing to make a choice, hopefully either choosing the appropriate antibiotic the first time or understanding if this is viral or a bacterial infection, so that hopefully we can reduce the total number of antibiotic treatments that are needed.
And so is your role in that team to decipher through doing, bioinformatic analysis, for lack of a better term, who's there, who's causing the problem? Would a traditional farm send you samples and say, hey, can you help us figure out what this is?
So our goal is to be able to our goal is to be able to let the farmer do this all on their own, or a veterinarian.
Sure.
That we can develop this, basically the decision making part of it, or the recommendation part of it, to make that all automatic. But right now we are sorting through and trying to determine, so we've actually created a large data set. We've collected samples from almost 1,000 cattle from 6 states.
Wow.
And then we've determined how abundant are the 4 most common bacterial pathogens. And we're also quantifying the most common viral pathogens, and then trying to determine at what combination of these pathogens will then go from a healthy state to a sick animal.
Yeah.
And I'll just say one little thing is it's complicated because most of these bacteria are present almost all the time, but at a level, either the animal's immune system is such that it can ward off the infection, or there's not enough of the bacteria to cause a problem, or maybe the proper combination of virus and bacteria hasn't caused an infection. So it's really difficult to, but we're getting better and closer at being able to predict what a sick animal looks like and what a healthy animal looks like.
That's fascinating. And yeah, I think you touched on a point there. I guess if we can finish off with some of that, is the quantification aspect. I know a lot of your publications have looked at who's there, but how many of those are there? And perhaps even, as you were talking about, their metabolic contribution, what's happening there. There's this field of metagenomics, I guess, that is exploding, and it's allowing us to look deeper. Can you touch a little bit on that? What's that bleeding edge of knowledge with metagenomics, with quantification, using the modern genomic tools and sequencing tools that we have?
Yeah, so metagenomics is the process of taking a sample and basically extracting all the DNA from that microbial community. We'll focus on microbial community. So take a fecal sample. There are billions of bacteria in this little, maybe 1 gram of feces. Extract all the DNA from it, all the DNA from all the bacteria, and then sequence everything with the potential goal of sequencing every genome of every organism in that sample. That's been allowed by the advance in DNA sequencing technology, which has reduced the cost of sequencing about 10 million times cheaper today than it was 30 years ago.
Wow.
Which is a huge number. And it's not an exaggeration. It's literally 10 million times cheaper today than it was 30 years ago. So we're still, usually this results in generating about 10 million sequence or 100 million sequences in a single sample, which sounds like a lot, but there are billions of bacteria in a sample also. So we're not sequencing everything, every organism in a community, but we are able to get a pretty good idea of the entire genome of the most abundant bacteria in communities. There's a lot of technical problems that we're still working on, but the goal is to be able to take a sample and then determine the complete genome. The complete genome is the entire, all the DNA in an organism for as many of the bacteria as possible. And I think that's going to continue. I think probably something in the future that's going to happen is to try to make this a little bit more targeted, because there are so many bacteria. Developing methods of isolating single bacteria and then sequencing those maybe individually, so the current method is to extract this whole big mixture of DNA and then just sequence everything and then solve the puzzle and pull it all apart afterward. That's okay. But I think we find that disentangling this puzzle of which DNA belongs to which organism is still a limitation. So I think it's my opinion, in the future, a little bit more organization of and sorting out and separation of the community up front, and then applying big sequencing to it to simplify the puzzle afterward is going to be a future area in metagenomics.
So interesting. So, and I'm sure machine learning and artificial intelligence is going to help us with that as well. Tim, is there anything that we didn't talk about, or any question that I didn't ask you that you would like me to ask you, or something you would like to talk about to finish off this wonderful interview? And thank you so much again. You've been a wonderful teacher, and it's such a great discussion so far.
Boy, I feel like we really hit all the best hits. It's been a lot of fun. I think just the take home is yeah, hopefully we can all appreciate and respect our gut bacteria a little bit more. We can feed our own bacteria by eating lots of fiber and maintaining our intestinal health, and appreciate farmers, appreciate the animals and the food that we have. I think it's we take for granted, but definitely there's a whole lot of research, a whole lot of work, and a whole lot of care to get us the food that we need.
Absolutely. Great words to end this, and great words of wisdom. Thank you so much, Dr. Timothy Johnson, Assistant Professor in the Department of Animal Health, Animal Sciences at Purdue University. But we're talking about animal health. I feel inspired to give you a new department as well. But thank you so much. It's been really revealing, and I love how you took us through this journey and opened up my eyes. Definitely, I hope people are also listening to some of these words of wisdom and taking care of their animals, taking care of their own guts as well. And thanks again, Tim.
Thanks a lot. It was a lot of fun.
Have a great day.